Our next guest is unfortunately not live with us, but we are delighted that she recorded her talk with us beforehand. After the session, she will be available to answer your questions. Her career and achievements are impressive. Her work spans from fundamental materials modeling to industrial application. She has published over 120 papers, holds 14 patents and 3 provisional patents, and has received various awards for her work. Today's presentation is about PHAS. PHAS are known since decades. Since the number of producers grew, advantages like marine degradability are one of the key known features of those. We are looking forward to learn about advancements she did at Queensland University. A warm welcome to Professor Laycock with a keynote speech on the topic PHA advancements, polymer modification, blends, composites, and end of life.
Good morning, everybody, and hello from Australia. I really do thank everyone for inviting me to this Congress and giving me the opportunity to talk about our work. So what I wanted to cover was a bit of a broad overbrush of aspects of biodegradable polymer research, particularly focusing on the work we're doing in our ARC training centre for bioplastics and biocomposites. So this centre was established a couple of years ago, and it's focused on PhD training. It has a number of industry partners, and it has around 19 PhD students and 5 postdocs with a focus on bioplastics and biocomposites, covering the entire gamut from synthesis to end of life, and with aspects of policy. So this is a summary of our structure, our centre structure. And as you can see, we're seeing it as a series of interconnected themes. So the key themes are starting off with more of the policy and the social license to operate work. That includes areas like life cycle assessment. It includes understanding social behaviour and some of the business and policy aspects around bioplastics and biocomposites. We then have another theme, which is biomass transformation. And this is looking at conversion of biomass into materials that are of use for further conversion down the track. So a large amount of this work is around some of the natural resources we have in the Queensland context. And again, we have this biopolymer production. So in this case, there's a large stream of work around sugar, which is one of the main industries in the Queensland context. So the sugar to biopolymer conversion program, for example. So actual synthesis of PHA in collaboration with the Queensland government and likely to go to pilot. And then we have a very large stream of work, which is around novel materials development and biocomposite production, biopolymer production, novel materials production. We also, within this framework, have our end-of-life work, which is biodegradation and understanding lifetimes in the environment, as well as some aspects of recycling. So as I said, we have 19 PhD projects that are started within the centre. It is actually a $13 million centre, including $7 million cash, and we have 5, not 4 new postdocs. But overall, the theme of this is advancing Australia's transition to a greener plastic future. And we really are a transdisciplinary group. We do cover everything from chemical and materials engineering to polymer chemistry, polymer science, environmental science, social science, policy, and business. So if we're looking at the centre structure, you can see here that we've got the 4 themes with a little bit more elaboration. So the upstream bioengineering, polymer chemistry and engineering, and we have lots of polymer biocomposite processing and products. So I'm going to be spending a little bit of time over the next number of slides, first off talking about some of the associated research programs, and then diving a little bit more into the aspects of the work within this centre in particular. But the background to all of this, as everyone would be very well aware, is the exponential growth in plastic production, of which only 9% is recycled, which is leading to a massive increase in plastic waste, which is entering our environment, our soils, our waters, our air. And this massive plastic waste problem is asking for multidimensional solutions. So what I'm showing in this slide is an elevator view of all of the types of work that we've been doing that start with the synthesis end. So in this case, we're dealing with materials such as purple phototrophic bacteria that take carbon dioxide and convert them to biopolymers. We're looking at methane to biopolymers. And then we're also looking at polymer synthesis for areas such as block copolymers, so polyhydroxyalkanoate-based block copolymers. And then we move on to other types of areas such as peptide self-assembly into conductive nanowires, a carbon fibre production from some of these materials, lignin-based polyurethane foams, and then a whole suite of materials where we're looking at composite production from various waste resources, starch hydrogels, including highly hydrophilic type materials for nappy type applications, and then a massive suite of work where we're looking at ultimate fate, so recyclability, biodegradability, lifetime estimation. So one of the parallel programs is the Food and Beverage Accelerator, which has just started up within our school. And this particular program is looking at premium food and beverage and looking at things like extrusion processes, high moisture extrusion, tailored vegetable proteins and so on. So textured tailored vegetable proteins. This area of work is a fairly intensive program within the centre led by Jason Stokes. We also then have another parallel program which is about to start up, hasn't yet started, which is the Plastics Waste Cooperative Research Centre. So this centre is focused on many aspects of solving plastic waste. We're a participant in this program, and it's working across areas including recycling as well as alternative material development and design of collection systems. So that's briefly touching on two of the associated research groups, and now I'm going to touch a little bit more on some of the specific projects within the centre itself, past and present. So one of the previous projects, it's no longer current, but we were looking at conducting peptide nanowires. So we were trying to do self-assembly of fibrillar peptides using aromatic groups that would have pi-pi stacking to give us conductivity. And this is a biomimetic process mimicking the nanowires that are associated with some bacteria that are used for conductivity even in aqueous environments. And so this was one of the early stage self-assembly pieces where we were actually designing from the ground up for polymers from novel materials. This particular project here is starch-based materials for plastic packaging and hydrogels. Within the centre, we have a new stream of work where we're looking at alternative materials which will have very good oxygen and water barrier properties. Starch has excellent natural oxygen barrier in its own right, but also we are looking at reactive extrusion processes where we can tailor starch to give us very, very hydrophilic cross-linked hydrogel materials that can swell 200 to 300% of their initial weight and act as very good moisture traps. So those projects are currently working in parallel. So the main focus for us at present is on starch as thermoplastic starch as extruded films and understanding and manipulating the properties of those films for commercial application. Another material that we're really focusing on a lot, and I've mentioned this already a few times, is the bacterial polymers, the polyhydroxyalkanoates. So these bacterial polymers are a very high focus of interest for us because they have some very unique properties. The primary property of interest is that because they are naturally bacterially derived and found in hundreds of native bacteria, they're also naturally biodegradable in a way which many synthetic bio-derived and biodegradable plastics are not. So these materials, these PHAs, will biodegrade in marine environment, soil environment, anywhere where bacteria pretty much are present, including anaerobic environments. So that's one of its their primary benefits. They're also thermoplastic, which means that they can be readily processed in drop-in technologies, and they are hydrophobic, so they're water resistant, which is a very important property. So if we're looking at some of the approaches to PHA synthesis that we've adopted, one program of work which has been done in the past, it's not current at present, was methane to PHA conversion, where we found that through techno-economic analysis that we needed to do thermophilic processing in order to avoid the costs of having to do cooling in the process, which was more than 50% of a cost of a plant, as it turns out, because of the exothermic nature of the biomass conversion process. So methane to PHA conversion was one strategy we were adopting to look at high volume starting materials resources for PHA production. Another that is current is the use of purple phototrophic bacteria, where we have high carbon efficiency use without CO2 emissions, because it's actually absorbing CO2 in the presence of hydrogen to convert to biopolymers. So these materials use natural light, infrared light, as the energy source for driving this process. This is early stage research, and we're still exploring productivities, but it is very interesting to see that these native materials do naturally accumulate quite high levels of PHAs. Sugar to PHA is another area of research that is very, very intensively being investigated at the moment, with pilot plants under design. One of the things about this process is that we're finding that extremophiles, so halophiles, are particularly useful for this process, and we're hoping to find that we can do native extraction using osmotic shock to help accelerate this and reduce the costs of this exercise. Another area of work that's very interesting for us is actually the chemical synthesis of block copolymers. So in the past, we've tried to manipulate the processing of alternating feeds to the bacteria to produce block copolymers, but that gives us a mix of products. It gives us both blocks as well as blends. And so what we're now doing is that we're starting from a base material, so the pure PHBV, which is a PHA, which can be purchased. We're using reactive extrusion to break it down to smaller blocks, and then we're rebuilding the structures with different blocks of PHA to give us very, very high elongations compared to the starting material. So we're going from around 7% elongation up to about 160% elongation, and very tough materials, which are suitable for packaging type applications using this strategy of block copolymer synthesis. And you can see that we've also been able to film blow these materials in our pilot film tower. This is actually a process which I won't be talking about the details of, but it is effective, and we are able to produce materials that are tough and flexible. Another thing that we're looking at is the use of hydrogels for ammonia capture and wastewater treatment. So this is a process where we're using, in this case, not the starch hydrogels, but related materials, which are very effective in taking up ammonia from wastewater treatment plants. And so we can use this as a way of capturing the ammonia before it enters the plant and saving a lot of energy and some of the processing post this stage. So this is a conceptual lab-based product where we have just done some initial exploration and found that we get pretty reasonable capture in 5 to 10 minutes of around 80% of the ammonia in the stream. A large activity for us over many years has been this concept, which is quite circular, which is the PHA-based wood plastic composites. So in this case, what we're looking at is taking organic feedstocks and through biomass fermentation, producing volatile fatty acids, which can then be used for the production of PHAs. And you can then blend those with wood pulp or wood fibers to make composites up to 50 weight % or more. And those can then be used for bioproducts like tree guards or medical trays or anything where you can see them as more rigid type products. This is a drop-in replacement type material. So this has a lot of attractive qualities to it because the use of the wood reduces the overall cost of the product, but also this is a truly circular type of process because, again, with the presence of wood, it's 100% biodegradable. We've spun that out to another project, which we've done preliminary investigations on, where we're looking at a wide range of types of feedstock. In this case, paunch, which is the kind of material that's present within a cattle rumen. So you can take that fibrous material that's semi-digested and actually can make reasonable composite products from that. So we've explored a number of different types of fibres, not just the wood fibres, but other fibres in these PHA composite products. Another advantage of the PHA is that it is, because it's very, very hydrophobic in its own way, but also biodegrades naturally layer by layer, it has a lot of useful properties for controlled release applications. And so we've done controlled release across a range of different types of products. One of the ones we're currently looking at is fertilizers, and controlled release of fertilizers, controlling nitrogen loss at the source, trying for higher nutrient use efficiency. And this is something where, with the coating of PHA, we're finding we can delay release of urea, for example, very significantly, from days to weeks, timing to match plant demand. This project is still, we have done extrusion processing for the production of these as blends, as matrix, as well as spray coating for the production of these as films. And we've gone to plant pot trials as well as open field trials with these materials. We've also looked at nitrification inhibitors and controlled release aspects there. And it is quite a complex release process. It's not simple layer by layer. And what I'm trying to show in this slide is that you can look at initial release as well as fractures and fragments and channels and pores, and then dissolution and migration as well. So multidimensional aspects of release, more complex than we initially thought, and we have to take that into account. And this shows you, for example, where we've mapped and monitored the changes in voids and the free volume over time with release, as well as looking at the urea concentrations to do models of the overall efficiency and release of nutrient over time, so that we can understand and predict and model alternative materials. This is another example where we've used slow-release biopolymer composites in control of pimelia poisoning in cattle in Australian outback. So this is a major problem for cattle. They eat this plant pimelia and they're poisoned by simplexin. And so what we're trying to do is trickle release of this toxin in order to give the cows immunity to this poison, with building up some resistance over time. And we found that these materials are very effective at release of active agents in model systems, but also we've done trials in the actual cattle rumen of PHA, just the biopolymer itself at this stage, but it is effectively degraded in the rumen with no adverse effects. Then, of course, we have the end of life. Plastics end of life, we've both got alternate fates, so looking at repurposing, catalytic depolymerization, low pressure PET conversion, carbon fiber production, a lot of aspects of alternate fate of biodegradable polymers at end of life, which is one aspect. And the other aspect is a big focus on biodegradation. So we do have real-time degradation in the field for our biodegradation studies, as well as laboratory-based respirometry, so we can monitor true conversion, mineralization, to carbon dioxide or methane. And both of these are working in parallel so that we can understand how these systems behave, get some sense of modeling their lifetimes, predicting their lifetimes, but also understanding some of the environmental factors that are going into their degradation. One of the things to understand about biodegradation is that you can have either surface or bulk erosion, but biodegradation is effectively a hydrolytic process. So it's water-based, and its hydrolysis is driven either enzymatically or abiotically, depending on the environment and the type of polymer. So PHA is enzymatically hydrolyzed, and it's many orders of magnitude faster than abiotic hydrolysis. Materials like PLA, abiotically hydrolyzed under ambient conditions, and they need to get above their glass transition temperature in, for example, compost conditions, in order for their rate of biodegradation to be reasonable. So it's either a bulk erosion or a surface erosion mechanism, and those two are quite distinctly different. One, the surface erosion, you retain material properties until you get quite thin and brittle before you fragment. but you lose mass. And with bulk erosion, you lose mechanical properties much earlier. You're basically hollowing out internally, losing mass, molecular weight internally. And the ultimate fate is carbon dioxide and or methane and water. One of the things to notice is that there are very different types of biodegradability for the different types of polymers. And so we do focus a lot on comparative studies where we're looking at, for example, biodegradation of PHA versus PLA and PBAT, and in some cases polycaprolactone, with reference to polyethylene. And these will degrade very differently in different environments. So it's always necessary to be conscious of what type of biopolymer it is that you've got. We have done multiple field trials, and one of the most recent ones is a massive field trial out in the environment, which is just wrapping up now. Publication's about to go out, where we looked at the effect of processing, and so solvent casting, melt extrusion, injection molding, as well as 4 different types of polymers in 4 different locations, as well as surface and benthic. And those results are just going through analysis at the moment, and we're getting some very, very interesting and distinct differences between the polymers. So one of the things we have considered briefly, and I'll only touch on this briefly, is whether or not toxic additives, either sorbed or extruded with the PHA, affect the biodegradation. And the very interesting result when we're looking at phenanthrene, which is a common toxic pollutant, or dibutyl phthalate, which is widely used as a plasticizer. So dibutyl phthalate at 20% did accelerate dramatically the biodegradation of PHA, and even the phenanthrene unexpectedly did accelerate after initial lag the biodegradation of PHA, and we don't entirely understand this at this point, although we have characterised changes in communities which seem to be associated with that. Overall, the conclusions for us with all of the work that we're doing, and we have published a paper on this in association with Cause, one of the major legal firms in Australia, is that biodegradable plastics do present a potential solution to some of the issues relating to plastic wastes. The challenges that have been identified have been that the scope and role of biodegradable plastics in an increasingly circular economy, they're not straightforward. We do need a multifaceted approach, and we do have this discussion paper where we've attempted to address many of the issues, particularly around labelling, particularly around legal frameworks for introducing these materials into a context such as the Australian economy. I'd like to leave it there, and thank you very much for listening to the talk. If you have any questions, please feel free to contact us at our website, and I'm looking very much forward to having the discussion after this.
Thank you very much for your great lecture, Professor Laycock, and a warm welcome to Australia. Hello, can you hear us? Good afternoon.
Yes, hi, I can hear you. Good afternoon. Yes, good morning to you. Good afternoon from here.
Good morning, good afternoon, and thank you. Great that you're with us. We would like now to open the session for questions from the audience. So does anyone have a question?
Robin Schumacher from the Center for the Transformation of Chemistry. You told us that your institute, your research center there is focusing on PhD students mainly. Aside that, are there programs where you raise awareness for biopolymers, say for younger students or even students in schools?
Yes, so we do some high school engagement and other engagement, but it's not a main activity for us at the moment. We're mainly focusing on our PhD students and our postdocs in the centre. We do make an effort to do a lot of communication, so we do media, and I have done quite a number of public talks to the community. So that is an exercise, but in terms of schools, it's a little bit limited.
I mean, there was a kind of huge focus on PHAs for sure in your teams. I know that here in the audience we have also lots of people with experiences on PHAs, PHBs. So maybe there's some question coming from that guys as well. So please feel free to stand up and ask some questions if there's so.
I have another question, not on PHA in general. Do you have, let's say, molding equipment in your a university that the material that you produce or research can be molded in extrusion molded or injection molded. Do you have this kind of equipment there as well?
So we've got film tower, we have injection molders, we have extrusions, many extruders going up to quite large scale. And the injection molders also go from very small to almost pilot scale. So we have a very well-equipped polymer processing laboratory. We're very lucky that way.
Okay, so you can transfer your results right away to the practice. That's very good. Thank you.
Yes, we've actually made material up to pretty much commercial trial level through the extruders that we have available to us. So we've been able to go all the way to making, say, 100 trays, 50 trays of a product for testing. So we're capable of that level.
Okay, thank you very much.
So yes, hello. Thank you very much for your nice presentation. And you pointed out that PHA and PHB, for example, are real biopolymers, directly produced by microbes. So
Yes.
what is your comment on the productivity? I mean, you must compete with other synthetic biopolymers where the productivity maybe is much higher. So what is your comment on that? What is the perspective of these materials?
So any biotechnological process is inherently always going to be slower than a chemical synthetic process. I would note that from the synthetic perspective, it's extremely difficult to synthesize PHAs. So you need very, very expensive specialist enzymes. And it's only in the last couple of years that you've been able to get the molecular weights that are obtainable naturally. So at present for these particular polymer types, the competing synthetic approaches are not particularly viable. In terms of competing with other synthetic approaches, obviously biotechnology is a slower process. It inherently will take time. But the scale does allow us to work as basically parallel batch.
Okay, so you also plan to scale up this production to bigger scale, industrial scale? Okay.
Yes, so at the moment, I can't talk much about it, but there is a pilot plant being developed within Queensland related to PHA production.
Okay, thank you very much.
Yeah, hi, good afternoon, and thanks also from my side for this great overview. With regards to end-of-life solutions, circularity, are you convinced that PHA products at the end of their life cycle can be a new feedstock for a kind of biochemical recycling to bring back with all the byproducts and biomass that you has shown to us to make new pellets and then bring it back to new products, and what must be fulfilled for that?
Absolutely. Look, it's thermoplastic, it's meltable, it's reprocessable. It's also very, very easy to take it back to monomers. So we've done a lot of reactive extrusion over time. So we can degrade different molecular weights in a very controlled fashion, or you can take it all the way back to starting material. And of course, there's circularity via biodegradation CO2 as well, which is the ultimate cycle through biomass. But it's not as, you can't go through as many cycles of recycling as you can with, say, polyethylene, but it certainly can be recycled multiple times as a ground material, but also it can be broken down to starting materials that can be used for building blocks. And that's what we've done with some of our block copolymer synthesis.
Okay, thank you so much, Professor Laycock, for your great lecture, for answering the questions, and for joining us during the Congress. We are wishing you a great day and great Congress. Enjoy. And
thank you very much again for the invitation.
Yes. Goodbye to Australia.